Assalamualaikum warahmatullahi wabarakatuh. My name is Umi Syafiqah Binti Ahmad Fahmi from AS201A2, and I will be presenting my FYP project titled DNA extraction and PCR optimization for amplifications of RAPD-OPH12 from oil palm samples. In this study, oil palm was utilized, and specifically the species Ileus guineensis, or known as African oil palm, that was originated from Africa. Obtaining highly pure samples of oil palm genomic DNA in sufficient amount is important to start an Ileus guineensis DNA analysis experiment. In this study also, mature leaves were used as it were the only available type of leaves present for sample collection. The genetic marker that was chosen in this study is RAPD-OPH12. According to a research article by Pasaribu, RAPD-OPH12 has highest polymorphic fragment compared to other primer. Thus, it was the best marker to be chosen for this study. The expected band size from this primer is 573 base pair to 2153 base pair. A successful DNA profiling depends on the quality of DNA template and optimized PCR condition. From sample preparation through thermocycle optimization, there are numerous elements to take into account. Amplification reactions require expensive materials and reagents, and PCR kits are not cost-effective in labs with sparse infrastructure. Thus, understanding each component of the reaction and adhering to a step-by-step protocol are ideal ways to obtain optimum PCR result. The objectives in this study was to observe PCR products of Ileus guineensis using direct FTA card and transdirect PCR method, and to observe relation between PCR optimization and formation of PCR products using primer RAPD-OPH12. For methodology, we are determined to observe the PCR products of Ileus guineensis using RAPD-OPH12. Firstly, we start with DNA extraction. We used two different kits, which were FTA card and transdirect PCR kit. For FTA card, 5 samples were ground and transferred to the card and punched out to be used. While transdirect PCR kit, 5 mg of 8 oil palm samples were cut and added into a tube. The DNA extraction steps continue according to manual provided with the product. Then we proceed with screening of sample of oil palm band size using primer EGSHP to ensure that the sample was from oil palm. Then, for optimization of PCR, we check the primer concentration, reaction reagents, and the thermocycle condition, such as the annealing temperature. Then, we proceed with PCR amplification with optimized PCR condition. To view the PCR products, 1% agarose gel was prepared and PCR products was detected using gel electrophoresis. These are the PCR components for both FTA and transdirect PCR kit and the PCR conditions. For discussion, we also run prior PCR experiment using primer EGSHP to ensure that the samples that we are using is from oil palm samples. According to research article by B. Kalyana Babu, the expected band size of Ileus guineensis is 550 base pair, and from 5 samples taken from FTA card, the result of PCR amplification shows the supposed band size, proving that the sample was from oil palm. For experiment 1, PCR1 from FTA card, temperature gradient was done from 35 degrees Celsius to 35.8 degrees Celsius with 0.1 degrees Celsius increment. Sample from well A shows DNA amplification as band at expected band size was showing at range 500 base pair to 2153 base pair. Though it's not as clear, it falls between 500 base pair and 600 base pair size ladder. There were formation of primer smearing, and it was expected because the annealing temperature from well 1 to well 7 was too low. For the next PCR experiment, PCR2, sample from well 1 to well 5 shows DNA amplification as band formed as expected band size range as shown in the picture. PCR conditions was optimized, and learning from experiment PCR1, the annealing temperature was set at 35.8 degrees Celsius, and there were amplicons showing. Because transdirect method has been done, the result is presented in experiment PCR3, and no DNA modifications was produced, and the reason was concluded that there were not enough DNA present from sample used, and the DNA concentration is less than 50 nanogram. To conclude this project, PCR products of Elais guineensis using direct FTA card and transdirect PCR method has successfully been observed. From findings from this experiment, FTA card were reliable as it produced enough DNA concentration needed for PCR analysis. And it is important to follow step-by-step protocol and do PCR optimization to ensure the success. Primer optimization includes primer design, such as primer concentration need to be maintained low to avoid primer dimer formation. Template sequence must be enough for this study. It was 50 nanogram. Next, the reaction reagents must be maintained at recommended temperature and avoid freeze-thaw cycles to maintain its quality. Thermocycler condition, especially the annealing temperature and extension time, must be maintained. It is recommended to be extra careful during washing steps of FTA card as DNA can be washed away and reduce DNA concentration needed. And this concludes my FYP project. Any questions and inquiries are warmly welcome. Thank you.